Welcome back from the break, everyone. In this session, we will be hearing from 4 additional speakers, and we'll again have a panel discussions where you can ask your questions. Please be sure to enter your questions in the Q&A chat pod so that we can have productive panel discussions. We will be starting off joined by John Henry, Director of OPQ's Office of Program and Regulatory Operations. In his time at FDA, Mr. Henry has managed pre-market and post-market applications as a regulatory project manager in the Office of New Drug Quality Assessment, now part of OPQ, and has facilitated the Quality by Design program. We will next hear from Dr. Ee-Sunn or Joanne Chia, who is Director of the Division of New Drug Products 3 in the Office of New Drug Products, or ONDP. In her current role, Joanne is shaping culture and helping modernize FDA's regulatory review operations in ONDP via numerous working groups and initiatives. This includes, most recently, the Knowledge-Aided Assessment and Structured Application for Drug Substance Working Group, which developed a knowledge-aided structured application framework applicable across both generic drug and new drug API assessments. Our third presenter, Joel Welch, is an Associate Director for Science and Biosimilar Strategy in OPQ's Office of Biotechnology Products. He is responsible for assessing emerging, complex, or precedent-setting issues impacting science policies of the office with particular emphasis on the biosimilar program. He also serves as the rapporteur of the ICH revision to Q5A R1 and the Vice Chair for the Emerging Technology Program. And our final presenter for this section will be Alex Viehmann, Director for the Division of Quality Intelligence 2 within OPQ's Quality Surveillance. This division performs post-market quality-based assessments of drug sites and products enhanced by data integration and analytics tools developed in support of monitoring and improving drug quality to inform congressional inquiries and data calls, future GMP inspections, enforcement decisions, and application assessment. Speakers, the floor is yours.
Good afternoon. I will discuss the integrated quality assessment and in particular the aligned team approach recently implemented by OPQ. First, I'd like to highlight pharmaceutical quality. A quality product of any kind consistently meets the expectations of the user. Drugs are no different. Patients expect safe and effective medicine with every dose they take. And pharmaceutical quality is assuring every dose is effective, safe, free of contamination and defect. This is what gives patients confidence in every next dose that they take. For my presentation, I have the following learning objectives. I want you to understand the IQA from a business and project management perspective. I will give you a recap of our assessment process prior to the IQA and then our assessment process after the IQA. Also, I want you to understand the benefits of the aligned team approach that I previously mentioned. And lastly, I will provide some of our best practices associated with communication during the application review. So what is IQA, or Integrated Quality Assessment? Think of it as having multiple assessors with very different expertise coming together as a team, doing collaboration to provide a single quality voice surrounded about the application review. Also think of it from the standpoint of a review and inspection. consolidating the review of the application with the inspection activities to form a single consolidated decision. Lastly, but definitely not least, as I mentioned in the previous bullets, collaboration and communication amongst the team members. This collaboration is very important in order to form this one quality voice, but also it enhances knowledge management. You have individuals that are working together, sharing their experience, sharing their knowledge, so that this information can be shared across different application types as well as across different disciplines. This is a pictorial of what the IQA would look like. You have different disciplines. You have your drug product, API, or drug substance, manufacturing, biopharm, microbiology, biology, all these different disciplines sort of working together to form this one quality voice. Prior to IQA, there were 3 different offices involved in the assessment, the quality assessment that is. You had the pharmaceutical science. They had the different disciplines, whether it was biology, microbiology, API, drug product, that looked at the application and reviewed that application. And then you had Office of Compliance and Office of Regulatory Affairs that did the inspection. With that, there were 2 separate sort of recommendations, an inspection recommendation and an application recommendation that went to the stakeholder, meaning it went to the Office of Generic Drugs or the Office of New Drugs, depending on whether it was an innovative product or whether it was a generic product. And with that before IQA, there were some concerns, right? There were assessors that were responsible for potentially large portions of the CMC module that could potentially have to review both drug product, drug substance, but may not have the expertise in both those areas, but had to learn those. As I mentioned before, the review and inspection was evaluated separately and in different offices, so that the synchronization of activities were very challenging. For instance, you could have a situation where the application review may have been closed out and the inspection has not even started, and therefore there wasn't the communication of things that could be done during the inspection. Or there could be situations where the inspection starts and maybe the review has not caught up with it. So that lack of synchronization could cause gaps in the need for collaboration between the inspection and the review. Also, the user fee programs are very siloed. New drug, biologics, generics, in the AA and the ABLA were very siloed, and therefore there was potential for inconsistency in assessment when there was need to be some consistencies. For instance, maybe in small molecule, where the science are similar, maybe there could be inconsistencies because there wasn't that collaboration and they were done things in silos. Additionally, with that, policy development could be delayed because there isn't collaboration when there needs to be the collaboration in some cases and harmonization across the user fee programs. And without that collaboration, some things can be delayed. So, what brought about the IQA? Why did we decide to go with and move forward with the IQA? So there were, with the CDER mission, and I know you've heard this from Dr. Woodcock many times, about how our mission has evolved and how some of the things have become more complex and some of the challenges that we had. For instance, drug shortages have started to come to the forefront. There are new novel technologies coming forth There's been some complicated products and incidents that happen, whether it's heparin, whether it's NDMA, even though NDMA was discovered probably fairly recently, the incident had occurred well before that. So some of these challenges that come up make us realize that we need to also evolve. And these challenges warranted these type of changes. And that created this whole formation of OPQ and had us think about unifying all the quality functions under one organization. I want to talk about, very high level, the idea and the concept between the concepts about OPQ and IQA. One of the bases is the expertise of the assessment, right? We want expertise-based. We want people that understand very logical, the expertise, whether it's drug product, drug substance, biopharm, people that understand that and have very expertise-based. That enhances the overall quality by having people that having the right people review the right portion of the application at the right time. So those people have that expertise. And then there is the team-based assessment, right? That you want these individuals now work together in a collaborative fashion to provide an overall assessment. Even though there are expertise, there is a need to collaborate because there are, in some cases, some overlap in the things that we need to review, and there's also knowledge that can be shared across the different disciplines. So that team base and trying to get that one quality voice is very important to have that collaboration so that we can communicate effectively, communicate efficiently, because there are timelines that need to be met. So that team base allows for the efficiency of our process. And then with that, there is the implementation that's needed, and that implementation requires a lot of training of the staff and development of the staff. We also required some tools that need to be developed. We developed the IQA, we developed different IT systems that facilitate our assessments as we move forward. Sort of a repeat of one of the previous slides where we're showing the disciplines, but in this case, one quality voice going to our stakeholders. So some of the things I mentioned before, what are the benefits of strictly IQA? Collaboration and communication, right? You have a very informed decision when you have the right expertise working together in a collaborative fashion. Two, by doing that, you have uniform standards that's going to be utilized. So as I mentioned previously, with that knowledge management, you have people sharing ideas, and then therefore from application to applications, their consistency. From policy and guidances, their consistency. We just continue to build that knowledge base within the organization so that we are consistent and provide very informed decisions. Again, quality assessment after IQA. We implemented this across, as I said, the ANDA, NDA, and BLA programs. I think for a high level, we achieved a lot of our goals. We had a one quality voice. We increased some consistency across the user fee programs, and we improved some collaboration. However, I think there are some improvement that's needed in a couple areas. We recognize that risk-based assessment could be improved, and our next presentation will go over CASA and how that has been incorporated. And then teamwork optimization, right? Even though we worked as a team, there still was a need to improve that collaboration and I'll talk about that shortly with the IQA aligned team. So, what is the IQA process? Right? You have a project manager, you have all the disciplines, and you can see in here, there are a lot of RBPMs, a lot of different individuals in each discipline. So how do you come about and get a team? You have an RBPM, but then you need your team, right? So you have a large pool of individuals, and each application requires a drug product, drug substance manufacturing, and then you do this for every application. And in doing so, you create a team every time, and you can have very many permutations of team members that you work with, but never working with a single unit. You're working with a new group every time. So that loss of collaboration on a consistent basis was not realized in the original IQA. However, with the aligned teams, what we did was have a smaller pool of assessors, and therefore you only work with a very few drug substance, drug product manufacturing, different disciplines in a smaller pool. So you start to build that relationship, start to build that knowledge within that team, and be able to communicate better. So this just provides sort of just, information around why and how it's better to work as a team as opposed to individuals, right? That a team-based review is much better than individuals doing work, because that collaboration is better. Even though with the original IQA there was some level, by having different teams every time, you just didn't get that consistent collaboration that you would get when you truly know and work with someone consistently. So, what are some of the overall benefits of the IQA with the aligned team? When you're working with the same people all the time, you can schedule more meetings. You can schedule meetings to talk about these scientific timelines and scientific discussions and timelines that you couldn't do as easily when you have multiple individuals that you have to work with. It allows better synchronization of your review and communication to the applicant. You're working with smaller people. We can synchronize things a lot better with our review. When people have different priorities because they're working on different applications, it becomes challenging to synchronize things. But when you're working with the same people all the time, it's easier to understand their workload and to balance the work appropriately. Also, with that better communication, it starts to eliminate some of the redundancy that you might get or the gaps that might happen when we're sending out deficiencies. So again, overall collaboration just makes for people working together, discussing things, finding out if there's any gaps, finding out if there's overlap, but at the end of the day provides a much clearer, concise communications, including clearer, concise deficiencies to the applicant. So what I want to also sort of pivot and talk about some of the best practices for communications. Your RPPM or your project manager is your point of contact. It's very important to utilize this person to provide communication. They are the central person to getting information that you need, answering questions. Oftentimes we've seen where communication has been sent to others, and which really delays, one, the feedback that you would get back, the response, but also it just creates a lot more effort on our part to get you the same answer anyway. So the RBPM is your single point of contact. Utilize that person to the best you can so you can get efficient and effective communication back. And then utilize pre-submission communications. This isn't available for all types of applications, but when they are, for instance, IND meetings, pre-ANDA meetings. Utilize that information, that venue, to get information about the application, get our feedback prior to submission. It can be very helpful when moving forward. Cover letters. Cover letters is our tool for understanding what's in a submission. It helps us to triage the submission so we can understand who needs to be involved, whether we need a consult, whether everything is there or not. Having a detailed cover letter is very important, and so therefore we ask that provide a high level of detail what's in that submission through that cover letter. It's a huge tool for us. Respond to IRs by the due date, right? So we generally give a certain time frame for that. It helps us schedule-wise, and also to provide meaningful feedback. Whenever that can't happen, though, again, the project manager is your point of contact. Utilize that person to the best of your ability. Our IRs, or our CRs, if that's the case, any deficiencies should include 4 parts, right? It has what we actually reviewed, what's provided in the application that we found deficient, what is that deficiency or issue, what information we need to resolve that deficiency, and why we need it. So those are 4 parts. If those parts are not there, feel free to reach out to get better understanding or clarity around your deficiency. But you should respond with what is needed. Read that part and provide that information, or provide a rationale for why that isn't there. If you just provide something and it isn't what we want and there's no rationale, again, it just has a circle of going through IRs. Right? Again, review the IR deficiency upon receipt. Contact your RBPM if you have any questions, comments, or concerns. And again, from a communication standpoint, utilize any post-action meeting requests when appropriate. This also helps provide some clarity around any communications that we may have had. So, in summary, I want to mention that the IQA, one, improves, the IQA provides an improvement based on expertise and teamwork. Expertise enhances the overall quality. The teamwork enhances the overall efficiency. They both kind of do both, but that's what they focus on as far as the expertise and teamwork. The aligned team continues to enhance the collaboration and yields a more informed decision. And then, as I talked about some of the best practice, being very proactive in your communication and transparency enhances the overall process. I think that's 2 ways, both us, meaning the agency, as well as the applicant. Being proactive and being very transparent just will make things go a whole lot better. So that concludes my part of the presentation. I will now like to turn it over to Joanne Chia, who will talk to you about CASA.
Good afternoon, everyone. I'm Joanne Chia, the Division Director of the Division of New Drug Products 3 and the Office of New Drug Products within OPQ. Today, I'm kicking off CASA Part 1, Modernizing FDA's Regulatory Assessment, and will then hand off to Joel to cover CASA Part 2, a talk on the BLAs. FDA's pharmaceutical quality has evolved over the years. We've developed various initiatives. Some of these include the Process Analytical Technology or Emerging Technology Program, the PAT-ETP, Quality by Design, the Integrated Quality Assessment, Concept of Operations for Facility Evaluation and Inspection of Human Drugs, and most recently, OPQ's Quality Initiative, known as CASA, which stands for Knowledge Aided Assessment and Structured Application. I show here on the left the ICH M4Q Common Technical Document Triangle, which provides a harmonized structure and format for presenting quality information in the CTD. Typically, our quality sections are within modules 2 and 3. Over the years, what we've noticed is that we've sort of evolved and adopted a read, summarize, and evaluate approach, where we follow the CTD triangle closely. We document our assessment in Word. We archive in PDF, which generates a really lengthy document, and it's often not searchable and appears to be a barrier for knowledge management. And at the same time, we're also faced with the expectation to achieve more consistency in our assessments. So FDA recognized the need to modernize our regulatory assessment. One goal is really to move away from the narrative or unstructured data to structured data, which would enable us to best capture and manage our knowledge. This is all possible because structured data is highly organized and formatted so that it's easily searchable in relational databases. And meanwhile, unstructured data doesn't have any defined format or organization, which makes it difficult for us to collect, process, and analyze. So CASA is part of the larger CDER effort to modernize our regulatory assessment. Now, what exactly is CASA? It is an advanced IT system for regulatory review and knowledge management using structured submission data in OPQ. There are 3 main pillars of CASA depicted here. Pillar 1 pertains to the assessment of risk to quality using established rules and algorithms. Pillar 2, this represents the assessment of the product design and the quality standards and how the risk is mitigated. Pillar 3 is about assessing the manufacturing process and the facilities involved and mitigating the risks that appear there. So CASA really enables us to leverage the structured application, our knowledge base, and drive a knowledge-aided assessment which we utilize for knowledge management throughout the product lifecycle. So why do we want CASA? The objectives of CASA are to deliver a structured regulatory assessment and submission, institute rules and algorithms for risk management and decision-making, perform intelligent data analysis to establish regulatory standards, and capture and manage knowledge during the life cycle of a drug product. Here's a quick look at the CASA interface and how we accomplish our defined objectives. CASA takes in structured application data and captures critical assessment information in a structured format. OPQ's integrated quality assessment for NDAs, ANDAs, and BLAs rely on expertise from various disciplines that include the drug product, biopharmaceutics, manufacturing, and drug substance. And altogether, this drives our one quality voice. So keeping all these disciplines in mind, OPQ developed CASA in a systematic, stepwise approach. So you can think of this as all parts of a bigger puzzle. Drug product, for example, we first introduced CASA for drug product, and then we developed CASA for biopharm, moved on to CASA for manufacturing, and most recently, CASA for drug substance. Now, although we have all these different flavors of CASA, so to speak, each CASA is based on the same fundamental approach of 1, firstly identifying the initial risk, and then secondly, assessing whether or not there is appropriate risk control or risk mitigation. When we look at CASA for drug product, the initial inherent drug product risk is based on the drug product characteristics. The corresponding product risk control focuses on the various aspects of the drug product that include the drug substance characteristics, the drug product design and understanding, and control of the CQAs. CASA for biopharm relies on biopharmaceutics-based risk assessment decision tree. The drug product risk control and mitigation here has a focus from a biopharmaceutics perspective. So for example, we consider the dissolution methodology, including the discriminating ability and testing conditions. And then we also consider the type of dosage form and whether it is an immediate release or modified release drug. Now, CASA for manufacturing, in this case, we identify the risks associated with the facilities and the specific unit operation. And then the corresponding risk control and mitigation relies on an assessment of the specific unit operation, and then we also factor in the facility or site capability and cGMP history. CASA for drug substance is developed for both NDAs and ANDAs. We have here an algorithm to calculate overall risk for predefined drug substance risk categories that can impact a drug substance CQA. The risk control here from a drug substance perspective, we consider the proposed synthetic pathway, the drug substance manufacturing process, and also the associated controls. So the key take-home message is that CASA applies the same fundamental risk identification and risk mitigation in a structured format across all disciplines. This is the key to promoting consistency and objectivity of the assessment. So given our limited time, I'll walk through only some specifics on CASA for drug substance and then illustrate how we achieve the objectivity. Now, for example, here, from a drug substance perspective, there are defined risk categories impacted by the CQAs. The initial calculated risk is based on preset questions. The answers are associated with FMECA scores of probability of occurrence, the severity, and the detectability to assess the impact of a risk category on the drug substance CQA. This way, we're able to have the consistent, objective, and quantitative risk assessment. Now, during the assessment process, the reviewer will assess the information in the application and can overwrite the final risk ranking with sufficient justification. For situations that are deficient, the deficiencies are also clearly identified and aligned with the level of risk. Another advantage of CASA is the ability to harness analytics. Now, take, for example, CASA for drug substance. It enables assessors to leverage prior in-house knowledge. So, for example, we can easily view acceptance criteria that we've seen in the past side by side with a proposed control strategy, and then we can flag more scrutiny if the criteria is outside of what we have come across before. And here, CASA Analytics is helping us identify outliers in applications. CASA for drug substance encompasses an assessment of the drug substance synthetic pathway. This, again, is a structured approach. For each synthetic pathway, we look at the inputs and the outputs, which we utilize a chemical structures library. We then assess the control strategy, which factors in the synthetic process design, equipment, CPPs, and in-process controls. Other aspects will include the starting material, control of intermediates, and reagents used in a process. The structured synthetic pathway in CASA for DS enables us to search, to visualize, and leverage prior knowledge, and this in turn helps us improve overall efficiency and regulatory decision-making. Now, while I've only showcased the CASA for drug substance in the past few slides, we can have similar analytics for the drug product. Now, in this example here, we can view pending impurity limits against limits that we've come across in the past and determine if we need additional information to assess a specific control strategy. In the case of CASA for manufacturing, if we have a certain product that utilizes the same unit operation, we can view the risks associated for this unit operation against what we have encountered in the past. This benefits our regulatory decision-making and also is a conduit to investigators when assessing the risk for facilities. So in summary, the benefits of CASA are really twofold and positive for both the FDA and industry and patients. For the FDA, we modernize the regulatory review. We can gain efficiency, consistency, and objectivity in our assessment. We enable a knowledge management across DMFs, ANDAs, NDAs, and BLAs, and we can excel in regulatory action and decision making. From the industry and patient perspective, we set clearer communications, transparency, and consistency. We gain increased first cycle approvals. This is especially true in terms of generics. And we can move toward more affordable and accessible medicines. So I leave you here with a summary of our journey. We've taken a stepwise approach where we develop prototypes for drug product, biopharm, and manufacturing in 2015, 2019, and piloted CASA in the ANDA space. We've launched versions 1 and 2 from 2019 to 2020 for the generic solid oral dosage forms. And then this year, we've introduced Release 3.0 on a CDER IT platform. And then in the coming steps, we're looking forward towards the Release 4.0, where we will be incorporating CASA for drug substance, the liquid dosage forms, and in the new drug space, INDs, NDAs, and eventually BLAs. So thank you very much for your time, and I'll take questions at the end.
Greetings, my name is Joel Welch. I'm the Associate Director for Science and Biosimilar Strategy in the Office of Biotechnology Products in the Office of Pharmaceutical Quality, and it is exciting to be here today to talk to you about CASA and our approach to developing it for biological products in CDER. In terms of the learning objectives, we're going to talk a little bit about the why of CASA, and we've heard a lot about that already in terms of how it fits into the ecosystem of knowledge management in the Office of Pharmaceutical Quality. We will talk about some of the unique opportunities as well as challenges for biological products in CASA and how we're going to develop a system to accommodate that. And then we're going to talk about finally the general development approach. How are we going to go about building CASA modules for biological products that aid in our assessment practices in OPQ. Before we move to that part of describing kind of how we're going to build CASA, we need to talk a little bit about assessment within OPQ. You can see here the organizational chart for the Office of Pharmaceutical Quality, and there are two offices highlighted, each playing an equal partnership role in an assessment of a new biological license application. The Office of Biotechnology Products, where I reside on the left, is where much of the early development of CASA is being undertaken. However, it's important to note that the Office of Pharmaceutical Manufacturing Assessment also participates equally in assessment practices, and for that reason we need to ensure that the microbiology and facility assessment considerations their office evaluates are going to be accommodated within our system. In terms of the why do we need CASA, you've heard a little bit about this already in previous talks, but it bears repeating that there are both internal and external challenges that drive our need to develop this type of system. Certainly the volume of new applications and that crushing workload that we all feel of an ever-increasing amount of new molecules hitting the door that seems like the workload will never stop going up, that's part of this. Certainly, two, user fee program expectations, specifically the need to take predefined actions at predetermined times for those molecules drives the need to make quick, agile assessment decisions. Understandably, there are many stakeholders that expect high quality medicines to be in the hands of patients as soon as they can be. And for that reason, we need to develop systems able to help us make those types of nimble assessments. And then finally, all this occurs against the backdrop of breathtaking technological advancements. And we need to have knowledge management systems that are capable of accommodating them. In terms of internal challenges, our assessment practice is really driven around freestyle narratives, specifically writing a long sentence describing an assessment issue, summarizing the information from a dossier around that issue, and in some cases, even copying and pasting data tables to describe it. Not surprisingly, this practice is slow and inefficient. It's poorly designed for knowledge management and knowledge sharing, and it really is a manual process that drives the assessor to make decision based on the expertise and knowledge at hand. So all these things are things that CASA, as we'll describe in the rest of the talk, are going to help to address. These are the 4 key objectives of the CASA system. And you've heard about this a little bit already, but I think it does bear repeating, specifically the why of CASA, and also to highlight that this why applies equally for each one of these principles to biological products, just like the rest of the molecules assessed in the Office of Pharmaceutical Quality. So CASA really is designed to help us capture and manage knowledge rather than information. What are the things we need to grab hold of as part of an assessment, as part of the documentation? And then to establish rules and algorithms to facilitate risk, both its identification and mitigation, and then to communicate that assessment decision. And if we've done those things, we've not captured knowledge, we've established rules and algorithms, we can, where it makes sense, use computers to help us make preliminary recommendations for parts of a consideration of an application. And then finally, if we do all those things, we manage the knowledge, we establish the rules, and we use the powers of computers to help us make part of our evaluation, then we can provide an approach that's going to radically eliminate this text-based narrative we just described and really position application assessment for success going forward. So this brings us to our first challenge question. Broadly, CASA has 4 key objectives. How many apply to CASA for biologics? Answer A, 3. Computer-based rules and algorithms do not apply. B, none of them. They're too complex. C, 1, only the knowledge management portion is anticipated. Or D, all 4 still apply. And the answer is D. So I've told you why we're doing CASA. I've also told you how CASA is really the same across OPQ. Let's start with what does make biological products different. And I think the differences can really be described as a function of either the nature of the process, the nature of the data submitted, or finally, an understanding of molecular function and context. In terms of the nature of the process, for the manufacture of biological products, there really are a few low-risk unit operations. Each unit operation typically provides control of CQAs or many CQAs, many of which have real criticality about them, including, for example, viral safety or aggregation. There are frequently unique safety concerns. Biological products are typically made in cell culture. They're in living cells, and with that, there are unique safety concerns. For example, potential for adventitious agent contamination, the need to demonstrate viral clearance. As we think about the molecular function and context, mechanism of action is a key consideration as we think about that regulatory expectation of potency. But to understand mechanism of action, you really also need to understand target and distribution. So not just what does a molecule attempt to bind or block or target, but also where is that target located and how is it distributed across tissues. In some cases, there may well be more than one mechanism of action. An example of that could be what I've depicted here, a monoclonal antibody on the left demonstrating binding and blocking, and on the right demonstrating effector function of a cell expressing a target. So all those things drive us to really understand the nature of what a molecule does as part of this assessment process. In terms of the data submitted, process validation is an expectation in a dossier, and we see that as part of the submission already, and our CASA system therefore needs to encounter and accommodate this type of information. Methods within biological license applications the methods may monitor multiple CQAs, and frequently those CQAs may not be fully resolved. An example is here a charge variant profile of a monoclonal antibody. You see those acidic and basic peaks have beneath them many different CQAs, quality attributes that need to be understood to make an assessment. As we think about the interface between the nature of the process and what data we see, a lot of this is driven by small-scale models, where some of our process description is established based on these models, for example, with resin lifetime. As we think about process capability, spiking studies may be used to to drive some of our understanding and our expectations for a control strategy. You may see, for example, spiking of DNA or host cell protein. Platform validation is sometimes employed. We need to accommodate that type of information, for example, for viral clearance. As we think about both understanding the mechanism and the activity of a molecule, as well as what a process needs to do, we have the consideration for product-related substances versus impurities. The distinction here, are they retaining activity? So does that oxidation or does that deamidation of the protein, does that render it with partial or abrogated activity? And we need to understand all that to really assess the capability of the process, but also the control strategy for the molecule too. And at the heart of all of this is complexity. Proteins are heterogeneous systems made in cell culture, and all these factors describe the breathtaking permutations and challenges with describing the knowledge around a manufacturing process for a biological product. So how are we beginning CASA? So our first prototype module, and this module is a risk-based assessment module for drug substance manufacturing. It only applies to fed batch monoclonal antibodies and original BLA submissions. So why are we doing that? Well, first, the complexity for drug substance manufacturing is as much of the complexity in the application relative to the drug product. Secondly, fed batch monoclonal antibodies are really where a majority of our submissions are right now. Originals are an easy place to start. Certainly we're going to build towards a prototype that allows us to accommodate supplements and the life cycle later on. At this point, it does not incorporate the OPMA considerations I already mentioned, though I think we all acknowledge that's a critical part of this and building a successful CASA system. In terms of what this first prototype module is described to do, it's going to include process parameter criticality assessment, process parameter range evaluation, and it's also going to include key elements of the process that aren't characterized but that need to be described. For example, a chromatographic purification resin, right? We don't characterize a variety necessarily within a dossier, but we do have one that is used in the process, so we need to describe and capture that type of information. So what are the key features? What is this going to offer an assessor? Well, it's going to really allow an assessor to make a good risk-based decision where the data submitted by an applicant drives up or down a particular ranking, and really to use risk ranking based on assessor expertise and scientific consensus as our starting point to make these types of good risk-based decisions. There are going to be flags for assessment issues as they emerge and information requests that we send, really to help facilitate that primary and secondary assessor evaluation as they go through that iterative assessment process. We want to be able to therefore capture revisions that are made during that assessment cycle. And then certainly there needs to be an assessment and a conclusion, and since this is a standalone module, it needs to be able to generate a summary that we can incorporate seamlessly into the final assessment document for inclusion as part of the final record. Finally, I think we all acknowledge Q12 is where we're going. We want to be thoughtful and consistent with those Q12 concepts and designing towards building a system that really links to those types of ideas. This brings us to challenge question number 2. What assessment activity is CASA for biological products beginning with? A, all portions of BLA are in its pilot. B, fed batch drug substance manufacturing for monoclonal antibodies, both stability and specifications for drug substance and drug product, and D, drug product manufacturing. And the right answer is B, fed batch drug substance manufacturing for monoclonal antibodies. So how does it work? We've talked about why CASA needs to be developed. We've talked about what its objectives and its values are. But how is our first algorithm module going to work for drug substance manufacturing for monoclonal antibodies? You can think of it as a series of just pieces designed to ask the questions that we already ask as part of assessment. There being an initial risk assessment, there being characterization of a process, there being validation where we ask validation data questions, there being range decisions about what the final process will look like, and then finally a recommendation. And the key feature here is none of the questions we're flagging are new. These are assessment questions we ask now, beginning with what was studied. Is something missing that we think is necessary? As we move to characterization, what kind of data was submitted? Is it product specific or is it prior knowledge? And then, is it good data? Is there a gap? Is there a hole? Is the characterization data, complete? As we move to validation, what are the proposed ranges and what was studied? And then finally, do those ranges make sense and are they supported? And as we move to the recommendation, asking ourselves, what's the final summary? What's the final outcome? And then capturing all that knowledge that we need to capture as we go through this assessment process. So what are the final risk rankings? Are there issues? Are there precedents? And then building in some of the final other considerations like Q12 and established conditions. So where to next for CASA for biologics? We highlight here the drug substance unit operations 1.0, and I walked you through that algorithm a little bit. But there are other modules, and I'll show you a screen capture of the first one, the drug substance, and then the viral clearance here in just a moment. But this highlights really that there's a bunch of modules and a lot of assessment pieces that we need to be thoughtful of. And this is not a complete list. But you also see below some of the key outputs we're starting to think of, which is really what type of informatics questions do we expect our system to help us address? And again, these are still works in progress and not designed to be, 100% comprehensive, but they are designed to help us answer some of the uses and I think some of the power of CASA system as we build towards where we need to move going forward. I'm going to show in the last few slides some final screenshots of first our drug substance module or part of it, to just give you a glimpse of what it's going to look like. So you see here, this is the range evaluation page. CV at the top just denotes this is column volume. It's a rinse evaluation for a chromatographic purification unit op. And you see some of the key features. On the left, you see basic questions. Questions, again, we ask now. Is there a good characterization study? What ranges were evaluated? What is the final proposed operating conditions? And then you see on the right links to IRs. Have we asked questions? Do we want to build in that knowledge and that understanding as we moved on? And then finally you see a green graph button and then some comparisons of ranges that allows us to really give an assessor a visual assessment and to move us out of that unstructured narrative text into knowledge that's going to be able to be evaluated at the fingertips of an assessor. So I'm going to show you a second module now, very briefly, which is our viral clearance module. And here, again, just a prototype, still under development, but you see some of the things that this type of system is going to incorporate as well. Understanding not just what unit op it is, but where the data comes from. Is there modular platform approaches? And then trying to build in that knowledge and that continuity in some of the features we capture. For example, you see process parameters, which are held constant, which are scaled down. All these links and all these expectations are already established in ICHQ 5A, but we want to make sure we're building them in here too and linking them to the commercial manufacturing process where necessary. And then even incorporating which values were studied so we really retain that key knowledge as we go through the assessment process. So again, a second module just in progress, but it's so exciting I wanted to show it to you because it's really a great glimpse of where we're going. So with that, I'm at time. And I want to just pause and say, it's been a real honor to talk to you about CASA and describe some of the incredible opportunities it provides us for knowledge management and consistency. Obviously, our CASA journey is only now beginning, but it's an exciting one, and we think we're really laying the foundation for using the power of this type of system. I think the final thing to note is really that while CASA for biological products will have some unique elements to it, it really is designed to incorporate the same strategy, the same approach, and the same values that the CASA system for the rest of OPQ is describing as well. So with that, I'll just pause and say there's been tremendous work on CASA across all of OPQ, and I want to highlight some of the key folks that helped with this presentation and also highlight how much we appreciate the greater work of CASA that's going on across OPQ and why it's such an exciting time. So with that, I thank you for your attention. It's been a privilege to be here, and I'm happy to take any questions.
Hello, everyone. My name is Alex Viehmann. I work in the Office of Pharmaceutical Quality, Office of Quality Surveillance, where I currently serve as a division director. And I'm here today to talk to you about the Quality Surveillance Dashboard, which is a project that has been ongoing for the past few years that really enables more effective and efficient surveillance, getting us to the goal of a comprehensive surveillance program. What is the Quality Surveillance Dashboard? Quality Surveillance Dashboard first provides a framework for consistent and up-to-date evaluation of facilities. It integrates and governs facility and post-market product quality data from across multiple systems. So when you think of the volume of data we have about facilities and the variability when it comes to recalls, field alert reports, inspection history, products, etc., there is a complex history of each facility in some cases. And this dashboard enables and provides a single platform that brings together all of these data in a more effective and efficient manner. It allows quick searches for information about firms and enables quick comparisons. So if you'd like to compare certain facilities across product counts, recall counts, the types of recalls, information about their field alert reports, the Quality Surveillance Dashboard enables that. It allows users to navigate details about the most recent and up-to-date information about products, such as a breakdown of applications by application type, by status, by what role they play in the supply chain. Are they manufacturers? Are they testers? What is the inventory of products? What does the distribution look like around the route of administration or the dosage form? Do they manufacture products that are currently on drug shortages, on the drug shortage list? Do they have pediatric flag indications? And what information about storage conditions? It allows for tracking and trending quality signals within a product lifestyle, things related to MedWatch, field alert reports, and recalls, and linking that to distribution data for normalization. And really around being more proactive, right? Where do we see shifts? Where do we see spikes? Where do we see trends and in increases and decreases? The Quality Surveillance Dashboard allows up-to-date visualizations regarding those. It incorporates results of predictive analytics, for example, machine learning models on pharmaceutical quality effectiveness modeling and different metrics related to the PQS, around certain other product quality issues, as well as inspectional outcomes. It utilizes natural language processing to enable more efficient data mining, and these include extracting and mining through information around its establishment inspection reports, or 483s, which are very unstructured, text-heavy documents, which in the end leads to more efficient and risk-based assessments. And it better harnesses database resources by applying programming codes to systematically correct things like typographical or incomplete data fields. It allows for very effective data validation and informs data quality improvements and enhancements on the source data. How is the dashboard created? Well, it starts with integration. The dashboard and the system it operates on extracts structured and unstructured data, both from internal and external sources. So what do we mean by internal and external sources? Well, we currently manage internal data, as I spoke to before, about things like recalls and inspection history and field alert reports, the products that a facility manufactures, as well as external data sources from our collaboration with external parties like Dun & Bradstreet and other entities. Then comes data management, right? And this is one of the more powerful aspects, is that we really develop code and business processes and rules that, number one, extract certain information, that number two, clean and standardize those information, and then bring together and integrate with the other linked data sources. So when we talk about keying on an FEI, right, you can imagine how many different data sources are keying on that FEI. Well, how do we integrate those together, and how do we systematically do that to automate the process, right? These data are updating every day, right? We could get new information about a facility every single day. So we need to develop more standardized and automated processes that do this on a real-time basis, right? It allows for better organization, and then you can also bring in input from SMEs, right? How can you complement, right, and really enhance the automation and the different data sources that you're pulling information from? How can you bring in SME expertise to help even standardize that even more, and then automate that process, right? And then we apply different predictive and text analytic techniques, right? You know, we talked a lot about some of the machine learning models and the natural language processing techniques that we use to extract information and target certain elements, and I'll get into that a bit more in my presentation. And then swinging around into visualization and reporting to really create interactive and robust visualizations for reporting to stakeholders. So when our business partners are utilizing this platform, right, is giving them the most not only up-to-date and quality information, but interpretable, right, that they can quickly be able to see things happening within the life cycle, right? And then also the interactiveness, right? How can we make it interactive that would allow for further drill downs and more exploration related to a certain class of products within a facility or a certain type of problems that that facility is having? How do we enable that quick drill down? And then communication, right? Really around communicating everything that we're learning and all the decision making that's happening based upon these data and these analytics and these visualizations. How can we continually improve the entire process? Why was this so important, right? Why did we go through this huge investment into resources, software, hardware, as well as time, staff time to do this? Well, number one, the biggest thing was we found an ineffective use of time searching in multiple sources, right? You can imagine if you're trying to put together a picture of a facility, you're going into 8, 9 different systems, right? Each system has data that is structured differently, different classifications. When you look at the data table, different column names, the list goes on, right? So the majority of the time was spent cleaning and just bringing the data together, and you were left with a very small amount of time to actually digest that information and provide robust conclusions. Inconsistent methods for organizing data, right? We need better standardizations of how we did this. Use of static data that needs to constantly be refreshed and updated. This was a huge burden, right? Because you're only as fresh as the date that you pull those data, right? We needed to build in automation. We needed to build in that data integration and that data management process. Different assumptions about data that we needed to ensure consistent oversight and governance. As well as a lot of fishing for insights in unstructured data in these very text-heavy data sources when it comes to the text within a product quality defect report or an establishment inspection report, right? How can we be more strategic, right, and use available technology and innovation to target, to mine, to say what are the common themes happening here? So I'm going to walk you through an actual case study, an example of how one could use our quality surveillance dashboard. And again, these are screenshots, and I will try to complement the screenshots with some of the different capabilities. But here would be your welcome page of really when the user logs into the platform, you would be able to, it's a facility-driven platform. So if you knew the FEI number, or if you knew the firm name that you were searching for, or you wanted to know all the facilities that manufacture a type of product, right, an ingredient, all right, or if you knew the application number, the search capabilities would then start populating the different firms that meet those classifications, right, and you would be able to drill in deeper on that particular firm. The next thing, when we talked about comparison, right? One of the things that our users wanted the ability to do is have a quick cross-sectional comparison of facilities. So if you were going to an example would be if you're going to inspect facility 1 and you knew that this facility also had other facilities globally, right? That this firm also had other facilities globally. How can I do a quick comparison to tag all of those facilities to say, okay, what are some similarities and differences when it comes to the recalls, the MedWatch, the field alert reports, their OAI inspection history, the number of shortage products or application products that they manufacture? This table really allows that interactive cross-sectional comparison. Then moving in deeper. As you start going through the dashboard, right, you have those different tabs from the home page around products, right? The first one would be around products, right? And so now we would go in to be able to quickly see the inventory of products related to this facility, right? And it provides different summary statistics and graphs and visualizations around the information related to those products, right? So for this particular case study, again, we know that there are 40 different unique applications with 26 approved, 2 pending BLAs, 6 approved and 2 withdrawn NDAs, as well as manufacturing 4 non-application products where we're getting from registration and listing data, right? And then looking below, right? So you have a table with all your counts, and then you'll also be able to see a breakdown by dosage form or route of administration, right? And so you can quickly see that for this particular example, over 70% are injectable products. But what you don't see on this screen is the interactive capability. So if you wanted to quickly see, okay, well, I'm not interested in the injectable products. I'm interested into the tablets that they manufacture. Well, you would be able to hover over this tablet on the pie chart and click that, and your whole table, everything would update to reflect only the products that are tablet. That meet that classification. Similarly, based upon the route of administration, if you just wanted to see the inhalation products, you would be able to click on inhalation, and the whole product page report is going to update to only reflect based upon the user needs. And similarly, if you wanted to just, obviously I wasn't able to show everything, but if you wanted to see, low drug load products or products that have pediatric indications, there are certain filtering capabilities that would update this report from a very interactive perspective. The next graphic is what we call a waterfall chart, and this is really, we use for risk-based decisions, looking at the application load over time. So from, on your x-axis you have date, and on your y-axis you have the count of products, the overall cumulative count of products. And then for each year, what we're doing is we're tracking how many products are they net gaining or net losing, right? So you can quickly be able to tell and make risk-based decisions, about that facility. Are they taking on a lot of product load in a quick amount of time, right? And also being able to drill in, okay, if they're taking on a lot of product load, are they similar products? So you would be able to drill into those given years where they're taking on a lot of product load, a lot of new products into the facility, to really get more information around what those products are. Are they similar, right? Do they cross different dosage forms, different route of administrations? Are they very clustered into similar dosage forms? So this is really, based upon our business partners using this information to make decisions about facilities. This is a very great tool. Then what we also see under the product page of this report is to really define the supply chain roles across both drug product and drug substance that facility is doing, right? So we understand all the different products that they're making, right? But are they from a drug product, from a drug substance perspective, and then what role are they playing? So you can quickly see, okay, for the drug products that they are manufacturing at this, that are being managed within this facility, 34% of them, they're doing manufacturing. 31%, they're doing testing only. 19%, they're doing both the analysis, the testing, and the manufacturing, etc. And the same thing from drug substance. So you're quickly able to see the types of roles that this facility is performing on their given products. And again, at any point in time, you would be able to click into any of these to drill down deeper into, okay, well, I'm just interested in the APIs that they're doing testing only, right? They're only listed as the analysis. Well, I would quickly be able to click the 38% and my report would update, and it would give me, it would give me a line listing of the products. It would give me all the information about those products that I would require. Moving forward, another need that we recognize is really the supply chain mapping. And if you know your facility, right, you have, based upon that FDA establishment indicator number, right, that FEI number, we can map it to other associated FEIs based upon the application number, and then also based upon ingredient. So if you wanted to know who, for your given use case, right, who is the, if they're the finished dosage manufacturer for 3 applications for the same product, right? I would be able to quickly see, well, who are their API manufacturers, right? And then jump to those API manufacturers quickly to say, okay, well, what does their inspection history look like? Give me some more information about this. So it's really enables a quick and dirty supply chain mapping that we're constantly trying to enhance, right? This is something we're working a lot with our colleagues across OPQ to enhance. How can we better look at the supply chain and have it up to date, have it interactive, and have quick drill downs that would be able to give you the insights that you would need? Moving forward into inspections and compliance, how can we map, right, across the timeline for a given facility's inspections, right? So number one, looking across for the past 10 years, right? How many inspections have they had? How many of them were OAI, VAI, NAI, right? Do we see any import bulletins? Do we see any untitled letters, any warning letters, right? And really taking in those structured data and converting it into a timeline with a slider. So if you wanted to just see, okay, I'm interested in 10 years. Well, now for my next case, I'm only interested in the past 3 years. We would quickly be able to toggle that to give you the inspection timeline, right? And to give you that quick aggregated picture of what their inspections have looked like from an outcome perspective. And do we see any other cases? As I mentioned, import bulletins, untitled letters, warning letters, etc. And then on the bottom, what you're also seeing is the application of our predictive analytics to also, right, give us a prediction about the next inspection, right? If we were to go inspect this facility, what would we predict the outcome to be based upon different patterns in the data, right? The facility operations, are they a manufacturer, are they a packer, label, the product type, the product types, their quality defect history, right? This was a model that was trained upon all the available information we have, right? And it's going to give you a prediction of what the most likely outcome of the next inspection is going to be. Also, to quickly be able to get the text information around things like the summary of the EIR. So we're pulling these data and we're extracting the summary text from our data views to quickly be able to tell our users, okay, what did the investigator summarize for this given inspection that you're interested in, right? So you're quickly able to see the systems that were covered on the previous inspection, what the previous inspection was classified, what the current inspection resulted in. So in this case, a 2-item 483, right? Give you those 483s. What were the verbal discussion items with the firm? So it's extracting the text from the EIR in a summary, right? So that summary text is available. And moving forward into some of the more advanced things that we're trying to do is what we're trying to use is NLP to create and extract concepts, right? So on the left-hand side, you see these text topics are related to, okay, well, we want to know when the investigator is mentioning things around stability program. So we write code that incorporates SME expertise, right? That uses not only keywords, but phrases, distances between words. If and or statements, the list goes on of what the capabilities are, and then we store all that code, and we are constantly scoring the EIRs, and we're saying, okay, well, I know for this given facility, right, for the last inspection, here's what the investigator said about the stability program. Here's what the investigator said about data integrity. Here's what the investigator said about media fill, right? So you're quickly being able to extract and target the information that our users need, right? And this is just a small subset of the different text topics that exist. And you're able to quickly see those sentences, right? So those sentences are extracted out, and you would be able to refer to them to not only get a sense of what is the investigator saying, but what's the overall sentiment of what the investigator is mentioning related to these things like stability program, data integrity, production documentation, etc. Moving forward into our tab around MedWatch report, which is very similar to our information around field alert reports and the other product quality defects, is quickly being able to get a snapshot over time around the total count of MedWatch moving from 2016 or all the way to 2021. And you can map that by dosage form, so you can see injectables versus kits versus solutions. And then what you'd also be able to do is for any given year, you would be able to drill in deeper, right? To get, okay, well in 2021, I see that there, thus far, there have been 5 MedWatch reports related to their injectable products. Well, I could drill down very quickly, right? And see, okay, well in 2021, what does that look like? You know, we've had January through October. Have they all come in? You know, did we get them all in August? Have they come in sporadically? What were these reports? What was being said in the reports? You'd also quickly be able to drill down into you're only interested in a particular product or only a particular dosage form. All right, those capabilities are there. And on the right-hand side, right, is the type of defects. Right, is this a finished product defect? Is this a device defect, a container closure defect? Right, and again, these have been classified based upon SME rules, right, and that whole data standardization process that we talked to earlier. And you'd be able to quickly drill down into any of these items to get more information that you would want to visualize. So it really gives users the ability to visualize by dosage form, product defect, different other indicators, which then supports these more data-driven and risk-based decision making. And moving forward into the pharmaceutical quality system, well, what are we doing around the pharmaceutical quality system? One of the things that we've tried to use is innovative machine learning techniques to make predictions around certain pharmaceutical quality system effectiveness metrics. So, and we've tried to align these metrics with, the literature. What is industry utilizing to better, measure how effective their quality system is. What is ICHQ 10 referencing? So we've come up with a couple of different ideas when we talk about investigation time, percent repeat deviation, human error rate, CAPA retraining. So the result of the CAPA is retraining, right? So different indicators around the PQS. What we're doing is using different surrogate data we have available through product quality defects to track historically from year to year to year, right? What are these metrics looking like? And then make a prediction moving into the upcoming fiscal year, right? And then also to benchmark, right? So not only to track and trend and make predictions, but to benchmark and put percentile ranks so you can see where this facility stacks up against other similar facilities. So just another way that we use advanced analytics to give us a sense and better objective data-driven insights into quality and make that information available to our users. So in conclusion, the quality surveillance dashboard, when we think about it, it's bringing it all together. It's a function of data integration, data management, and really that concept of automation and boosting productivity. The application of natural language processing, machine learning, and predictive analytics to be more data-driven and objective. How can we use these techniques to extract data? How can we use these techniques to be more proactive in our signal detection? How can we use it to enhance future inspections, make future inspections more effective, more efficient? How can we use them to support both pre- and post-approval decisions? For example, the ICHQ 12 implementation. And then also, when it comes down to it, really facilitating more data-driven, objective, and efficient decision making. Thank you, everyone. I appreciate your time. I look forward to the panel.
Thank you for the great presentations. Welcome back again for our second Q&A panel. If you haven't had a chance to enter your questions into the Q&A chat pod, please do so now. We'll answer as many questions as time allows. Our first group of questions, of about 3 questions, will go to Dr. Henry. And here's the first question. Now that you have at least 4 reviewers writing separate reviews instead of 1 reviewer, what policies and practices do you have in place to ensure that the team reads each other's reviews and communicates effectively? And the second part of the question says, how is the voice of the drug inspector captured in this IQA?
Thank you for the question. For the first part, we have developed internal procedures that outlines the roles and the responsibilities of all the team members. Right? The team is facilitated by a project manager and a technical lead. They meet, meaning the full team meets regularly to discuss status of the application, and they discuss some technical issues associated with that. So that's a big part of the collaboration as well. And then additionally, to ensure things are moving well and going well, we do surveys, right? We solicit feedback from the team how things are going, is there improvements? So we're constantly looking at ways to improve and adjust, including collaboration throughout. Like I said, I think the detailed responsibilities in the SOP outlines it, but we also have things in place as far as meetings that ensure collaboration. Regarding the ORA, so for each application, the project manager will reach out to ORA to get a point of contact for the inspection part of that. This may or may not at this time be the actual inspector, because the inspector may not be assigned as soon as we get the application. But they then are invited to our team meetings and invited into those discussions. And then once that person is assigned, they would start to attend any meetings and discussion, be involved in that. So we make sure they are incorporated at the very start, at least the ORA, and then eventually once the inspector is identified, they are brought in with the internal discussions that we have. So I think that should answer the question.
Thank you, Dr. Henry, for responding to that question. We have another question for you. When you mentioned the cover letter, is this also needed on the drug substance API submission? And we're only asking since the drug substance can be used in multiple drug product submissions.
Again, thanks for the question. So yes, the cover letter serves is very important to us from really understanding what the content of a submission is without actually having to dig into the overall submission and do an assessment. So it gives us a better understanding what the content is, especially if it's an unsolicited amendment for an API or a DMF. That would help us understand what we might need to do. Additionally, we very often will see situations where, for instance, a new facility may have been utilized, but is not very clear in the submission or the cover letter. So those things will potentially delay us in making our assessment, whether it's a facility assessment or not, just will delay the process. We eventually will find that information once we review it, but it's just that overall, it makes for a more efficient assessment when the cover letter outlines the details of that submission. Thank you.
Thank you for responding to that question. We have one more question that just came in for Dr. Henry, and here's the question. With the efficiencies gained by the integrated quality assessment aligned teams, will the agency be able to shorten the current user fee goals for original application review?
Again, thank you for that question. So yeah, there are a lot of efficiencies that are gained with the aligned team approach. But I think when we're talking about decreasing goal dates, there are a lot other factors that go into that discussion. So I think it's best to have that addressed through our user fee commitment talks, which eventually actually had recently concluded. But good question. I think it's probably more appropriate to be addressed during user fee commitment discussions. Thank you.
Thank you for responding to that question. The next group of questions will be directed to Dr. Chia, and here's the first question. Is the CASA system up and running? If not, when do you expect it to become available? And what does this mean in terms of how companies submit drug applications? It looks like we may be having some connectivity issues with Dr. Chia. We'll let our tech team work with her, and we'll move on to our next presenter. And the next group of questions will go on to Dr. Welch. And here's the questions that just came in. What safeguards are you implementing to ensure that there isn't an over-reliance on the feedback provided by the CASA algorithms?
So thank you for that question. I think there are multiple different approaches we're taking to build in those safeguards. I think anytime you develop a new tool, there's always a balance that needs to be struck to ensure you don't rely on that tool too much. In terms of how we're addressing that in the biological products CASA, I think the first step is certainly learning from the rest of OPQ that's a little bit further along in their CASA journey and asking how they've addressed questions of governance and continual improvement of the system and trying to learn from some of their development plan. I think secondly, piloting is a huge part of this. And I think Joanne's presentation nicely showed the long CASA journey and how building in piloting time is a huge part of that development process. So we're going to be thoughtful about piloting and developing that part of our approach as well. And then I think finally, reinforcing to our assessment staff, it's still just a tool. And I think this is a place where, we're striking that balance of CASA, making some recommendations, flagging things that look a little unusual, but also that being a part of an iterative assessment process where a team leader, an application technical lead, as Don Henry described, provide some back and forth with an assessor to make sure they're getting an assessment decision right. So it's just a tool, and we're thoughtful about building a tool that fits our needs and really helps us make thoughtful assessment decisions. So thank you for that question.
Thank you for responding to that question. We've got a couple more questions for Dr. Welch, and then we'll be able to check back in with Dr. Chia. And here's the next question for Dr. Welch. How is the data imported into each CASA module, and how do you ensure data integrity?
So that's a really good question. I think one of the themes that came through the presentations you've heard today is CASA is part of a larger knowledge management initiative in OPQ. And to that end, other elements, for example, Q4M, PQCMC, are going to help, address part of the data input piece of this. But at the same time, CASA does in the interim need to make sure we have high quality data. So just like any other assessment, there is an oversight piece. There's an assessment of and a back and forth between a secondary reviewer and a primary assessor, and ensuring we have high quality data and that the data is right going in is something that process can address too. So we certainly want to build in the ability to have high quality data, and we're looking for, I think, a variety of different ways to address that need. So thank you for the question.
Thank you for responding to that question. We have one more question for Dr. Welch, and here's the final question. What can BLA applicants do on their end to enable a more facile CASA assessment of their applications by the FDA?
Well, I think what you've heard in terms of how CASA works is really it's about making high quality regulatory decisions based on the data submitted. So having a well-organized dossier, having a dossier where we can clearly identify the data you're referring to and building in links between them and making sure it's clear and transparent, I think is the most important thing. And then I think we also highlighted in presentations how CASA works, how there are flags and IR. So providing information when we request it and giving us the information we need when we need it, I think is part of it. But ultimately, these things aren't really new. You know, good assessments now have those elements to them already. So I think continuing to give high quality assessments and high quality applications is the most critical piece of ensuring that the CASA assessment works seamlessly and on our side. So thank you for that question.
Thank you for responding to those questions. We're going to check back in with the Dr. Chia and see if the connectivity is working. And here we are. Here's the first question for Dr. Chia. Is the CASA system up and running? If not, when do you expect it to become available? And what does this mean in terms of how companies submit drug applications?
Hi, thank you for the question. I hope you all can hear me now. Apologies for that just now. Yes, I mean, I echo a little bit of what Joel already mentioned before. CASA is a journey. Short answer to the question is yes, it is up and running. But the longer answer is that we've really taken a stepwise approach. We've developed prototypes over the last 5 years, and then in 2019-20 we released a couple initial versions of it, and most recently in 2021 we rolled out CASA 3.0, which includes drug product, biopharma, and manufacturing modules for generic solid oral drug products. We are using CASA to help us shift towards a structured regulatory assessment. It's based on built-in rules and algorithms for risk. And I think it's important to point out that, within the agency, we have multiple initiatives in parallel. CASA focuses on standardizing our assessments. There is no separate CASA submission. I mean, I think Joel might have mentioned before, there's a separate PQCMC, which focuses more on the structured submission. All right, thank you.
Thank you for responding to that question. Having a couple more questions for Dr. Chia, and here's the next question. Who decides which manufacturing step in drug product manufacturing is high risk or low risk? And does it depend upon the type of formulation?
Okay, that's a good question. So I think, so one thing to point out is that, the pillar of CASA is really assessing the control of risk. And CASA is based on the fundamental approach of where you first identify risk and then assess whether or not there's appropriate risk control and risk mitigation. For CASA for manufacturing, we actually have an intelligent algorithm capability which factors in the various unit operations. CASA for drug product incorporates considerations on the formulation design. So if the question of who is, there's an assessor who would go through the module, but it actually utilizes built-in algorithms. Okay, thank you.
Thank you for responding to that question. We have one more question for Dr. Chia before we move over to Dr. Viehmann. And here's the final question for Dr. Chia. Has the development and methodology underlying the CASA algorithm been published and the subject of peer review and or public comment.
Yes, actually, we have had input from industry and academia. And in fact, I think it was back in 2018, there was an FDA advisory committee meeting. We had unanimous support from industry and academia for the CASA initiative. The support we got was to work to enhance the submission format to improve the efficiency and consistency of the regulatory assessment. In terms of in literature, we also have had publications. We have one titled FDA's New Pharmaceutical Quality Initiative, Knowledge-Aided Assessment and Structured Applications. And this was published in the International Journal of Pharmaceutics in 2019. This paper was authored by Dr. Lawrence Yu, Dr. Susan Rosenkrantz, Andre Rowe, and other CASA experts within the agency as well. Okay, thanks, Ray.
Thank you for responding to that question. We're going to move on to Dr. Viehmann for the next group of questions. And here's a multi-part question. Is QSD only accessible to FDA employees? And what are you showing looks fantastic and it's very useful. Is access possible already now? And if so, how?
Thank you, Ray. So the answer to the first part of the question is yes. The QSD application is only available to internal FDA staff, and it's actually not available to all FDA staff at this point in time based upon the size of the environment that this is running on. So we're currently trying to scale it to a larger number of users, but as you can see through the presentation, a lot of the information we are assessing and managing are not publicly available information. So the answer to the question is yes, it's only accessible to internal FDA employees now.
Thank you, Dr. Viehmann, for responding to that question. We do have another question that came in for you. And here's the question. Who curates the QSD? Isn't there a constant need for entering massive amounts of new data into the QSD?
Thanks, Ray. Yeah, as everyone knows, the data quality is a major issue. So especially as we're setting up data pipelines for automation, there's a huge effort that goes into validating and confirming our data views are producing what we expect them to produce. So that's being managed both by a technical staff within FDA as well as OPQ staff that are actually writing the code and generating the data pipeline. So that's probably one of the biggest efforts in initially setting everything up is when we're trying to automate these data outputs is to confirm and validate the outputs are what we expect them to be. So that's currently being managed internally both by CDER technical staff as well as OPQ staff that's writing the code.
Thank you for responding to that question. We had one final question that just came in for Dr. Viehmann, and here's the question. How do you see the quality surveillance dashboard impacting surveillance moving forward?
Great question. The main impact we see is really efficiency gains. Historically going into multiple different systems to extract information and then bring that information together, the amount of time spent cleaning data and staging it in a way that it could be assessed only leaves a very small amount of time to actually process and digest and produce insights. So the main advantage is really efficiency gains as well as gaining insights from a lot of unstructured data, text data that we deal with. Is that we've built in capabilities to mine through this information and pull out and extract very critical information without a manual review. And this all leads to just more data-driven decision making from a surveillance perspective.
Well, that's all the time we have. Thank you to our presenters for all the great presentations and for answering numerous questions that came in. We'll now enter